All right, so yeah, thanks to Jasmine for doing so much of the introduction for me, because we're doing kind of similar stuff, but from a different angle, I would say. So I'm leading Plant Hologenomics Group, but I'm also not always around because I split my time with another institute based at Aarhus, but it's actually near Slough, so about an hour away, but it's super applied. So we have the fundamentals here. And I'll whiz through it because I've been a lot of conferences, so I know that many of you've seen stuff before. But I wanted to bring up basically that.
What is happening? Are you pressing
the arrow.
But I just did. Doesn't work. But we just checked that it worked.
Okay.
Let's try and see. Okay.
Now it works. Okay, good. So the kind of core idea was basically, can we improve crop microbiomes to make agriculture more sustainable? And obviously using hologenomics. That's why I'm here as well. And this has kind of been a long time, but I had some work in it's kind of stuff people have done a lot of, but with Daria, who's been around, and Maria, who's around here as well, that basically, like, if you take microbiomes from modern crops and wild crop relatives, they're different. And that's, 20, 30-year-old results. And we just did a small pilot study, but we took a bunch of wild maize, tortilla synths, and 2 modern crop varieties, and basically the 2 modern crop ones always have different microbiomes compared to the wild relatives across, like taken from across their natural ranges, even when you grow them together. But really, so what, right? Different genotypes have different microbiomes. What does that mean? And really, what's sort of been really exciting in the last 10 years, it's been a lot of emphasis on is trying to get some fundamental principles. So because things have different microbiomes, does it mean that they actually, is it random or is it actually having an effect? And actually, way back in 2007, there was a really nice study on exactly rhizobia. So we have these nice 3 groups in plants, the rhizobia, nitrogen fixing bacteria that Jasmine was talking about, and then arbuscular mycorrhizal fungi, and they help with nutrient uptake and stuff. And they really clearly benefit the plants. And what's really nice is that this study from 2007 was they took rhizobia, so the nitrogen fixing bacteria, and they found one that was really good and one that wasn't so good. And then they took a bunch of lines that were developed from bred from like 1900 to like 2000, and they basically just inoculated them. And they all got loads of growth benefits from the good rhizobia. But if you added a good rhizobia with a bad rhizobia, a poorer performing one, and you mix them together, over time they would get less benefits from the mixture. So that the newer crop types basically couldn't choose the optimal microbiome as well as the older ones. So actually, like, there's a decrease in the performance of the microbiome in modern crops. And I put together like a framework that I'm trying to work towards. But basically, you have these wild relatives that have just evolved over time with their microbes, with the environment. And you actually have these early land races, so the stuff that was spread by Mesoamericans in Mexico and stuff like that, where it's domesticated super slowly over time for a geographical area with the environment. And I think that they're actually showing the same sort of patterns as natural selection as the wild relatives. And then you get to like 1950s and you suddenly just go, all right, super breeding, loads of water, loads of nutrients, and you kind of decouple this relationship between the microbiome and the plant. And really, there's like 2 kind of competing hypotheses mainly. One is that the idea that you can actually select against having a good microbiome. So if you're using nutrients and energy to feed a healthy microbiome, if you suddenly don't need to do that, you can actually get high yields. We select for high yielding stuff. Crops you're actually selecting against promoting a robust microbiome. But the alternative thing is you just remove that selection pressure. And then because there's no kind of push for a constant optimization of these interactions, you can just have adverse alleles accumulate without negative effects. So that's really what I'm interested in testing. So there's evolutionary theories using the root microbiome, particularly using barley and maize, I would say. And so one of the things I also got what's called a Willem Young Investigator grant last year. So this is a chance for a PhD, who will be coming here for some of the time, I think, but to test the whole microbiome, the whole root microbiome across a whole bunch of cereal crops and their wild relatives, and actually kind of test the evolutionary pattern of the regulation from different environments, soils as well, and then also their drought response, and basically quantify how much benefits they get from their microbiome. And I really want to ask some questions like, do actually wild plants consistently get better growth benefits from their root microbiome? Even, for example, if you grow them out of their natural habitat in agricultural soils, for example. Also, can you use wild gene crops to actually identify the genes that promote really strong like plant microbe interactions? And finally, how specialized are microbiomes, right? So if you take a plant from a highland teosynthis or wild maize, and you grow it in a wild highland microbiome, it's actually getting a lot more growth benefits from that compared to, say, the lowland ones, which are, 2,500 meters lower down. So is that kind of, if you can quantify the growth benefits, then if that's actually specialized. But so there's a catch. So I have this concern. So with modern crops, and it's funny because I think just going to have this exact picture, but with the middle bit cut. But the point is you have massive changes. So on the left, this is the teosynthis, and this is kind of what it looks like. It's bushy, has bushy above the top ground, bushy root systems. The mod maize is super narrow, super dense. And then obviously the seeds are completely different. But the point is what happens if they're just too different? So apart from NNF spending 500 million Danish kroner on stuff that might not work, the point is they might just be too dissimilar, because if you have a gene that's beneficial in a wild relative, it might not work in modern crops. So it's also why we're taking another approach, which is basically using just modern lines. So even within modern lines, like you can see that the differences in performances and difference in microbiomes. So for example, here it's just a maize plant. One is super drought tolerant, one less so tolerant, but they both look like maize. If you sequence their genomes, they look pretty similar. They look very similar, but you can kind of easily cross them. And if you introduce one gene from one to the other, you have a good idea of what it would do. So that's why there's also, I have another project looking at these, and actually this barley project, barley microbreed, which I've talked a lot about already, but basically using a hologenomic approach to improve the drought tolerance of maize. So it's a super important crop for North African cultures, but also in Europe as well. It's known as the last crop before the desert. And basically you have some of them that are super drought tolerant, and others which are higher producing but more drought susceptible. So we can kind of cross these to kind of breed for better microbiomes and make their more drought tolerant. And so this is with Nikolai, who's over there, and then Aditya, who's now new and working on, so Nikolai is doing the genomes, and then Aditya will be integrating all the big data sets together. So we'll have a lot of data. So whole genome data, we have microbiome data, phenotype data of the roots, and then phenotype data in terms of the biomass. And we can correlate that all together, and hopefully we can find some genes which both affect the microbiome and also the drought tolerance. And hopefully there'll be something that we can then, and this is just to show we had a huge amount of like just digging in soil and planting stuff, but also then lab work and sequencing, and there's just a lot of integrating field and stuff together. And then basically, if you identify genes which you think both change the microbiome and the drought tolerance, you can actually just go in and play around with CRISPR-Cas on plants. Obviously, you can't use them, but you can test your theories, and you can see, does this both change the microbiome, and does it even affect the drought tolerance? And if you can kind of confirm a gene candidate, then you might be able to just breed it naturally or through these kind of breeding approaches. And then just to wrap it up, so yeah, this is the team, Aditya, Nikolai, Daria, she'll be back in next month for a few months, and just gets an honorable mention. He's part of this group, but he's working on tip hologenomics, but also random plant projects, which I can't say very much. So there you go, that was it.